Okay. So I guess we can wait a few minutes until people start coming in.
Sure.
Let's see. Yeah, I see people have started attending now. So hello, everybody who has just joined us. Yeah, let's give it a few minutes and 2 or 3 minutes until people start connecting, and we can slowly start. Okay, I see more people coming in. Hello again to the people who have joined us. Welcome. So I think we can start now. So again, hello to everybody who has just joined us. Welcome. It's a pleasure to have today Dr. Varun Venkataramani. And he will be giving us a very interesting talk about how to view differently brain cancer in a newly evolving field of neuroscience cancer research called cancer neuroscience. So before I give the floor to Dr. Venkataramani, I'm just going to go through a brief CV introduction of him, and then I will give the floor to him. So Dr. Varun Venkataramani is a group leader in the Department of Functional Neuroanatomy at the University Hospital of Heidelberg, and also a subgroup leader in the laboratory of Professor Frank Winkler, again at the University Hospital Heidelberg. He did his MD thesis developing novel methods for super resolution microscopy in order to characterize synapses. In his PhD, he discovered and characterized synaptic contacts on glioma cells under the supervision of Professor Frank Winkler and Professor Thomas Kuhner. And currently, he's working on the role of synaptic contacts in brain tumor networks and other cancer entities. Dr. Venkataramani, thank you again for accepting the invitation to be with us today. I very much look forward to your presentation. And the floor is yours. And for any questions, please type them and we can address them at the end of the presentation. Thank you. The floor is yours.
Yes. Yeah, thank you so much for the invitation. I'm very happy to be here today and to talk to you about the cancer neuroscience of brain tumor networks. And I'd like to give you a brief introduction about brain tumors, and we have no conflict of interest here. And just want to briefly start with the hallmarks of incurable diffuse gliomas, which are the most common malignant primary brain tumors. And they're characterized by the infiltrative growth and colonization of the entire normal brain. You can see on the right-hand side an autopsy section of a patient with an IDH mutant glioma stained with an IDH mutation-specific antibody. And you can see here in the brain, in a brown mass, the main tumor mass, but all the black dots indicate where you can still find tumor cells throughout the entire brain, which makes it clear that these tumor cells cannot be completely resected, and the remaining parts, the remaining cells are therapeutically resistant. All exact cellular and molecular mechanisms have not yet well been understood, while conventional molecular therapies have all been failures, latest in phase 3 of clinical trials, which makes it clear that the relevant biology has not yet been hit. And to really understand the underlying biology, we also need novel tool sets to really understand what the relevant biology is about in these tumors. And what we've been starting to do a few years back is to really look at these tumors with a methodology that is usually used in neuroscience. It's called in vivo two-photon microscopy, where we use a chronic cranial window and combine this with state-of-the-art patient-derived xenograft models that were fluorescently marked. And combining these two methods, we can really nicely recapitulate realistic growth patterns of diffuse infiltration. But we can also follow single glioma cells over time, over several days and months. And this is exactly what showed these results, where you could see that these tumor cells indeed show tumor microtubes, so basically membrane-like extensions, neurite-like extensions that allow these tumor cells to invade the brain, to colonize the brain, and to also form a tumor network where tumor cells are connected with each other. Using these as a framework, what we could see basically both, not only in patient-derived xenograft models of glioblastoma, but also astrocytomas and diffuse midline gliomas, that these tumor cells form these kinds of networks via tumor microtubes. But we can also see these tumor microtube networks in the human disease, in human diseased tissue, where we use mutation-specific antibodies or antibodies that stain tumor cells. So what's the role, what is the significance of these tumor-tumor networks that are gap junction coupled? And what we could see is that one of the important roles of these gap junction coupled networks is that they seem to mediate therapeutic resistance, because these tumor-connected tumor cells are, in the end, those that can survive radiotherapy, chemotherapy, and also surgical resection, whereas those that are not connected with each other are ones that seem to be much more sensible for or sensitive for therapeutic interventions. So this led us to the concept that these incurable gliomas hijack neural mechanisms. We also identified a lot of molecular drivers that are basically all involved also during neurodevelopment. And these microtubes again lead to the creation of functional tumor cell networks that again in the end also mediate cellular homeostasis and therapeutic resistance. And basically the start of my own work was to characterize these tumor microtubes. And when doing so, using high-resolution microscopy and also electron microscopy, what we could see is that these tumor cells are not only forming networks with each other, but they are also communicating with synapses. And we could always see that these tumor cells here marked in blue, so basically stained with a DAB precipitate, they are contacted by presynaptic boutons. And what you can see nicely here is that these synapses have all bona fide hallmarks of a synapse, synaptic vesicles, docked vesicles, a synaptic cleft, as well as a postsynaptic density. And when we then again use three-dimensional reconstructions of these tumor cells, what we can nicely see that predominantly also these synapses are formed on these neurite-like extensions. So another function also of these tumor microtubes. Again, we cannot only see these synaptic-like structures, synapses, neuronal glioma synapses in patient-derived xenograft mouse models, but we do also see them both in low-grade as well as in high-grade glioblastoma, showing us that this is not an artifact from the xenograft model. We do see different types of ultrastructural synapses, and again, as I already mentioned, we can see that these neuronal glioma synapses are mainly located on tumor microtubes. Now, the second question was whether these tumor neuron synapses are not only structurally there, but whether they are also functional. And for this purpose, we were able to use human perioperatively resected tissue that we then acutely slice. And in the living brain tissue still, we're able to patch, use patch clamp electrophysiology to patch tumor cells. And what we could see in these tumor cells is that indeed a subpopulation of these tumor cells showed EPSCs, so excitatory postsynaptic currents, showing again that this synaptic input that they get is also functional and can be measured in the form of electrophysiological currents. We could also reproduce this phenomenon again, both in the xenograft model as well as in co-culture model, where we culture tumor cells together with neurons. But we never ever see this the other way around. So we never see that these tumor cells are also the presynaptic side. They don't show any kind of action potentials reminiscent of neurons. So they are not differentiated neurons, but they receive neuronal input. They also do show another current that is much more enigmatic for us. It's what we call neuronal activities dependent slow inward currents. So we can inhibit these currents by washing in TTX as well as upper receptor antagonists. But we don't know really the role of these, and we don't know the molecular characterization of these currents. Neither do we know the tumor biological role. However, when we try to understand much better the role of neuronal activity, we also want to understand whether this has a functional consequence in vivo. And for this purpose, we used calcium imaging together with an optogenetic construct. So where we have infected neuron with an optogenetic construct. And as soon as the screen goes dark, we stimulate neurons and look at the calcium signals in the tumor cells. And what you can see is that after stimulation, that you can nicely see that the basal slow cancer tumor cells get much more synchronized and also much more frequent. So again, showing that also these slow currents, these EPSCs can get translated into calcium transients, into the functional communication in vivo in these cranial wires. So what does it have to do anything with tumor biology? And for this purpose, again, we used an optogenetic paradigm where we looked at tumor cells that are showing calcium transient as a response to neuronal stimulations and compared them to non-responders. And what we could see is indeed that the invasion speed of these tumor cells is highly correlated with neuronal activity. And also when we inhibit cortical activity with general anesthesia, again, this also inhibits glioma invasion. But we cannot put patients under constant anesthesia. So what is the clinical relevance? For this purpose, we used an FDA-approved upper receptor inhibitor, non-competitive upper receptor antagonist called perampanel. And we could see in the cranial window paradigm is that indeed the cell proliferation, glioma cell proliferation, significantly increases under control conditions, around twofold change, whereas those mice treated with perampanel show a static tumor growth, so not a cytotoxic effect, but a cytostatic effect. And interestingly enough, these phenomena that we see in adult glioblastoma could also be reproduced by the group of Michel Mongi in Stanford, and could see also these synaptic-like structures in DIPG, and could see that also perampanel has a significant effect on the proliferation index of these tumor cells. So taken together, this led us to the concept that there are always unidirectional synaptic inputs on glioma cells, always the neurons being the presynaptic side, always the tumor cells being the postsynaptic side. These tumor cells can get currents in the form of EPSCs or slow inward currents that again can get translated into calcium transients that drive proliferation and invasion of these tumor cells. So what we have done now, so what I just talked to you about are heterogeneous tumor networks. And we have basically two kinds of tumor networks. We have tumor networks, basically where tumor cells are connected with each other via gap junctions. And we have tumor neuron networks where there are synaptic inputs as well as perisynaptic and paracrine stimuli that play a role for driving tumor progression. So the question that we posed to us was, how do these different kinds of networks interact? What kind of role do these different kinds of connectivity cell states have for tumor biology? And the first observation we made is that there is even a third player in tumor cells that it connects with tumor cells, and these are the astrocytes, where this has been described before with in vitro settings. We could show that also tumor cells are directly gap junction coupled, not only with each other, but also with astrocytes, and that these connections are also functional, as seen here with calcium imaging. So using this dye, SO101, which can stain now not only tumor-connected cells, but also the connected astrocytes and the tumor cells, we can differentiate these different kinds of populations. So tumor cells that are connected with each other and or astrocytes, and tumor cells that are not connected with each other and astrocytes. And we can do two things. We can superimpose now the molecular cell states and the functional cell states of glioblastoma cells using this dye. So we can give this dye, we can use our cranial window mice model and differentiate those cells that are connected with each other from those cells that are not connected with each other. Look at which cells drive glioblastoma cell invasion. And on the other hand, we can use this dye to separate these two broad populations of tumor-connected cells and tumor-unconnected cells, and then perform single-cell RNA sequencing and integrate this with human patient data to come up with a model to understand how molecular cell states and functional cell states are related. And interestingly enough, we found that those tumor cells that are not connected with each other are the ones that are driving invasion. So you see the tumor cell here wandering around alone in the brain. The red color, you see blood vessels. Whereas those tumor cells that are connected, highly connected with each other, are stable in place, again, mediating therapeutic resistance. So again, showing that different kinds of tumor cell states mediate different kinds of tumor biological functions. Coming back to the single cell level, what we could also nicely see is that we can molecularly broadly separate unconnected tumor cells from those tumor cells that are connected with each other. And using a pseudo-time algorithm, we could at least pose the hypothesis that there's a natural progression from unconnected tumor cells to tumor-connected tumor cells over time during glioblastoma evolution. However, as you know, the pseudo-time algorithm doesn't allow us to infer any directionality. So which is why we again came up with the idea of understanding these types of connectivity with our in vivo model, and where we can see these tumor cells that are not connected with each other and do not take SN101 dye up in the beginnings of tumor invasion. But over the course of several weeks, we can see that these tumor cells take up this dye and get much more connected over time. Again, confirming the hypothesis that in the sparse infiltration zone, unconnected tumor cells drive tumor invasion, whereas this gets more stable over time in the tumor-connected networks. This again, we can also quantify on basically looking at how many tumor cells get connected over time, and again, confirm this hypothesis. But then again, the question is, how does this relate to the known classifications of single cell states by single cell RNA sequencing? And there are two broad classifications that we used and applied here. It's on one hand is the pathway-based classification by the group of Antonio Iavarone, where we could see that the unconnected tumor cells are those that are enriched for a neuronal-like cell state. And we used a gene-based classification from Mario Suber's group, where we could see that the unconnected tumor cells, so basically the drivers of glioblastoma invasion, are the ones that are enriched for NPC and OPC-like cell states. Again, showing that invasion seems to be molecularly mediated by a neuronal-like and OPC-like, NPC-like cell state. Now we wanted to understand whether these kinds of phenomena that we see on a molecular level can be also reproduced on a phenotypic level. And for this purpose, we again wanted to come up with an imaging paradigm where we can, with high temporal and spatial resolution, are able to follow cells in the course of minutes and to look at glioblastoma, tumor, microtube network dynamics. And we came up with an AI-based deep learning approach, basically here restoration microscopy, to really being able to restore the very fine processes of glioblastoma cells and follow them over time. And when doing so, what we could see is that these tumor cells perform very interesting phenomena that again are reminiscent of what neuroids are doing during neural development. They not only can branch out to really scan their microenvironment, they can protrude, but they can also retract. And so basically, again, these are the three basic mechanisms of what neuroids are also doing during neurodevelopment. On a more phenotypic level, what we could also see, and even using more high-resolution microscopy, we could see that there are even structures that are much smaller than tumor microtube. Basically, these small processes, they are very dynamic structure, filopodia-like structure. We do see TM-like structure that are much shorter than normal tumor microtubes. And we see lamellipodia-like structure, which has a much broader basis, and again, reminiscent morphologically of lamellipodia. And when we look at this from a more phenotypic level again, what we could see is that there are also, on a cellular level, we do see three different kinds of motion that can be also seen during from neurons, from immature neurons during migration phases. These tumor cells are showing branching migration, where the tumor microtube branches and then a somal translocation follows. Locomotion, where there's a protrusion of the tumor microtube and a somal translocation, and a translocation where the tumor microtube stands still while there's still somal translocation. These are all three basic mechanisms of neuronal development. Again, this is how it looks like within the cortex of the mice, where we can see that at least branching migration seems to be the predominant mechanisms within the superficial layer of the cortex that allows, where we are allowed to image with two-photon microscopy. But we also saw, as in contrast also to our previously assumptions, that these tumor neuron synapses only occur on connected tumor cells. We also see that indeed, even on invasive cells, which seems to be kind of counterintuitive to the stable notion of synapses, we do see transient synapses that are formed on invasive tumor cells. We do see here again also both with correlative light and electron microscopy, but also here with patch clamp electrophysiology. This again led us to the question, how does neuronal activity influence tumor microtube dynamics, tumor microtube genesis, and invasion speed? And what we could see again using our neuronal activity-dependent paradigm of general Dobson stimulation, we could see that the neurons where we stimulated highly showed that the tumor cells and the unconnected tumor cells are showing much more tumor microtube dynamics and invasion, whereas those that were kept under anesthesia were much more prone to not show any dynamic structures. So what are the mechanisms for this purpose? And what we could see is, again, looking at the much more high resolution three-dimensional calcium imaging, what we could see is that there are different kinds of calcium patterns, much more complex, much more reminiscent what is going on in glial cells, that we could classify as to the previous assumptions that there are only synchronized events of tumor cells firing together, but there seems to be a much more subcellular ultrastructural world of calcium transients within these glioblastoma cells, electrical active tissue. And we could see is that when we again inhibit calcium transients with a chelator like BAPTA-AM, we could see that indeed invasion, TM branching, and the small process genesis significantly decreased. And looking again at downstream mechanisms, what we could see, looking again at what kind of transcription factors might mediate these kinds of events, what we could see is that CREP, so basically phosphorylated phosphorylation of CREP, a clearly neuronal molecule, seems to also again mediate these calcium-mediated downstream targets. So again, coming back to our parent-panel paradigm, where we previously looked broadly at cell proliferation, here we also looked at TM formation and function. And what we could see again is that indeed parent-panel significantly decreases TM length, TMs per cell, as well as the branching points. So again, a subcellular level of mechanisms of action, how AMPA receptor inhibition and neuron glioma synapses mediate tumor microtube dynamics. So this led us to the overall concept that we can nicely see that really neuronal-like features mediate glioblastoma invasion on several levels, on the molecular level, where neuronal-like cells are enriched for the unconnected tumor cells that drive invasion. We do see on a phenotypic level neuronal-like cellular migration patterns. And we do see again that neuronal stimulation of the synapses leads to phosphorylation of CREP, to calcium channels, and then to the generation of new TMs, TM growth, and glioblastoma cell invasion. And this is again a nice framework how we can bring together the tumor biology of invasion together with the cellular and molecular heterogeneity. However, what we were only able to do in this study is to really look again at the cortex of the tumor cells. And again, we know that glioblastoma are predominantly a white matter disease. And so we were thinking about how to advance these kinds of imaging strategies. And for this purpose, we were looking at more of three-photon microscopy, where we want to understand whether we can also image these kinds of cellular phenotypes deep within the brain. And again, this is work that we did together here with the group of Robert Prewedel from the EMBL. And what we could, first of all, what we looked at were autopsy sections again to see how frequent also corpus callosum infiltration is in tumor cells, tumor patients. And what we could see is that in over 80 patients in these not so densely sampled corpus callosum samples, that we could see already here much more than 80% of invasion in the corpus callosum. And we do see this phenomenon in 100% of a patient-derived xenograft cells, where we have very nice microscopic resolution to observe this phenomenon, where we can see that all of these tumor cells always use the corpus callosum as their highway. And the question is, how can we observe the phenomenon that I just described also deep within the brain? And for this purpose, we came up with a workflow that allows us to combine adaptive optics three-photon microscopy together with an artificial intelligence-driven image processing, where we always have to struggle with low signal-to-noise ratios. Deep in the brain, we can make use of adaptive optics plus AI-based denoising. And we can already see here, again, this is the limitation of two-photon microscopy, where we only can image up to a depth of approximately 500 micrometers with two-photon microscopy. Three-photon microscopy allows us deep imaging up to at least 2.2 millimeters within the brain. And again, using adaptive optics, we are able to really nicely resolve cellular ultrastructure. And looking at this from an imaging point of view, we always have to deal with certain trade-offs for live cell imaging. And we have to always take into account whether we want to preserve the sample health and spatial resolution, temporal resolution, as well as the signal-to-noise ratio. And using, again, artificial intelligence, we're able to broaden this space up and make use of, to restore low signal-to-noise ratios. Using here, again, deep learning-based denoising, plus a removal of structured noise. Here using a 3D unit model. And again, being able to really preserve and increase the signal-to-noise ratio significantly. That allows us, again, to study the fine process dynamics, not only of the tumor cells, but also their microenvironment of the myelin and the blood vessels. And for this purpose, we made use of the third harmonic generation signal that, again, not only captures blood vessels and myelin, but again, when we use machine learning to really learn what kind of structures are blood vessels and which are myelin, we can nicely distinguish these structures and are able to image tumor cells, blood vessels, as well as white matters, in the tumor cells, and simultaneously to really understand how these structures interact. And what we can see when we want to look at this, we can nicely really go through the whole cortex. We can nicely, here again in the XZ projection, we can see the myelin here, the tumor cells in green, and in red, the blood vessels. However, we are not only able to do this from a microscopic imaging space, but we are also able to really nicely zoom in on single glioma cells, here in blue again, white matter, and green now the tumor cell. And you see the fine processes, these dendritic-like small processes, blood vessels, as well as the myelinated structures in the corpus callosum. So this allowed us now to really understand tumor cells and the microenvironment in much more detail. And we can see that indeed there's always a clear adaptation of tumor cells to the host microenvironment. We can clearly see that these tumor cells mostly align with the phylated fibers, while there's a subpopulation of tumor cells that seems to be even orthogonal to the myelin fibers. And we always were wondering how tumor cells can even enter this biomechanically very different environment of the corpus callosum. What we can see indeed is that they use blood vessel, perivascular migration along blood vessels to enter the corpus callosum. And this is much more frequent, again, in the corpus callosum than in the cortex. And when we look again at those cells that are orthogonally aligned to the myelin fibers, we can also see that there are two routes of perivascular invasion in the corpus callosum. Basically, one that uses perivascular-guided translocation, and one where we have, again, perivascular invasion, where the tumor cell is aligned along the blood vessel, and again, showing that this is the predominant way of how also migration orthogonal to the myelin fibers can happen. We can also confirm this again with correlative light and electron microscopy, where we can nicely see that the tumor microtube indeed touches the astrocytic end feed on the capillaries, and can see that this is not only can be seen on the light microscopy level, but also on the ultrastructural level. Again, we do see all the same phenomena that we also see from team dynamics that I just showed you in the cortex. However, we do see certain patterns of dynamics occur much more frequent in the cortex, such as branching, as well as retraction, whereas other mechanisms such as protrusions are much more predominant within the corpus callosum, again showing that there are probably different kinds of molecular mechanisms that underlie these processes that we cannot at the moment really, that we need to further characterize. We do see this again also on the live cell imaging perspective. We do see this also from a retraction side, and again also we do see branching even in the densely packed areas of the corpus callosum. Looking again at the cellular phenotypes that we just described, where we have these three types of migration patterns in the cortex, we do see the same phenotypes also in the corpus callosum, whereas in the cortex, the branching migration is the predominant one. This is much to a lesser frequency true in the corpus callosum, showing again probably that their tumor cells are adapting also their mechanism strategies to their microenvironment. And thinking about the idea of tumor-tumor networks within the corpus callosum, which is something that we were not really sure whether this also happens in the white matter, we can nicely see now that although the tumor cells are very aligned with the phylated fibers, these tumor cells still find each other and connect with each other in the densely packed white matter, showing again that this is a hallmark of this diseases. And lastly, what we can now make use of is, again, the capability to not analyze the tumor cells, but also the microenvironment in real time, and look at biomarkers of white matter reactivity in the corpus callosum upon early stage invasion and colonization, which is, again, clinically very relevant, because what we want to understand in the clinic is, when do patients already have white matter disease? To what extent does it also influence the white matter? And can we use any kind of sequence for this? And what we can see is indeed that we can make use of a biomarker of the cellular densities that are present within the glioblastoma, that they seems to be, at least from a shape-wise, seem to be significantly different. What was surprising for us to see that the directionality of the myelinated fibers does not change. So basically also, again, indicating that other mechanisms or other imaging paradigms, such as diffusion-based imaging, DTI, tractography, will probably not yield any significant results for these very early stages of tumor infiltration and giving us a clinical readout of early glioblastoma invasion. Now I want to switch gears a bit, because I was just talking about glioma. And just question is, for naturally that was occurring for us, whether these neuron tumor synapses also occur in brain metastasis, another focus from a group. And this, again, where what was known beforehand is that these synapses I was talking about on tumor cells occur, and that there are also perisynaptic contacts of both glioma cells, but also breast cancer, brain metastases, where it was shown by the group of Doug Hanahan that NMDA receptors seem to mediate tumor biological relevant macrometastatic colonization. However, it was not clear whether there are really bona fide synaptic structures in the very earliest stage of the micrometastatic cascade. And in work that I've been doing together with Mattia Garman, is what we saw is that indeed these tumor cells, these breast cancer brain metastases, show bona fide synaptic contacts on the very earliest stages. So you see here the capillary and one extravasated cell, and we can see again bona fide synaptic contacts by electron microscopy, both in breast cancer but also in melanoma patient-derived xenograft models. And from a functional side, we again see the same thing as also in glioma. In the earliest stages, we do see again EPSCs and also slow inward currents, whereas we never see any kind of action potentials. What does it again mean for the tumor biology? Again, here we made use of a functional AMPA receptor construct, where we made the functional AMPA receptors unfunctional with a dominant negative version of AMPA receptor. And we could see, and also here with perampanel, and we could see in the end is that the brain metastatic burden, the brain metastatic number significantly is decreased under perampanel treatment, and also with dominant negative receptor construct. And again, this we could see both in breast cancer brain metastasis as well as in melanoma brain metastasis. Again, leading us to the concept that there are not only synaptic contacts between neurons and brain metastases that are indirect, but also that there are direct AMPA receptor mediated contacts that are much probably important for early brain seeding, whereas those that are shown by Doug Hanahan's group seem to be more relevant for macrometastatic growth. So what are the future goals that I'm seeing that we need to really accomplish in the next years? And I think the future goals that we need to accomplish are that we need to understand the brain cancer connectome on a molecular, cellular, and physiological level. We need to develop and adapt methodologies across scales to study the cancer neuroscience, not only in model system, but also in human tissue. And we need to establish neuroscience-instructed cancer therapies as a novel pillar in cancer therapy. And for this purpose, what we need to come up with is really also specialized cancer neuroscience. So what I hopefully made clear is that we need to study these phenomena across scales, from clinical imaging up to the nanoscale level of electron microscopy. We need to study it in human patients longitudinally, not only during resection and re-resection. We need to study the treatment options that we have, but we also need to understand how it behaves after the death of the patients in the autopsy specimens. And we need to also obviously try to come up with surrogate parameters, be it clinical history, tissue, or liquid biopsies, and also establish appropriate ex vivo cancer neuroscience models. We need to integrate all these data in Cancer Neuroscience Hub, and then again, make use of reverse clinical translation to being able also to understand how does it behave from the preclinical side. Again, what can we investigate? How can we refine these therapeutic interventions? And again, I think the idea that I want to convey here is that there are not only tumor cell intrinsic mechanisms that we can target and broad mechanisms such as inhibiting proliferation with chemotherapy, but there can be a novel kind of tumor disconnecting therapies that we can make use of, where we use a disconnecting strategy of neuron tumor networks and tumor cell networks, where we can make use of neurocancer disconnection to establish these neuroscience-instructed cancer therapies. And with this, I want to summarize and give a brief outlook. So what I made hopefully clear today is that there are neuron tumor synaptic networks that drive glioma and brain metastatic progression. There are 3 layers of molecular and cellular mechanisms that drive glioma invasion, not only in the cortex, but also deep in the brain. That there's tumor network-specific targeting that offers novel therapeutic strategies and needs further clinical and preclinical investigation. And that it's important to create these interdisciplinary cancer neuroscience hub to ensure an rapid clinical translation progress of the field. And always the challenge of these interventions will be, if we think about these intricate mechanisms that are hijacked by these tumor neurodevelopmentally inspired tumors, is to preserve the integrity of central nervous system function while still having a therapeutic window to target tumor cells. And with this, I just gave you a very broad overview of what we want to accomplish. And again, if you are interested in this kind of topic, I just want to cordially invite you to attend the Heidelberg conference on cancer neuroscience that is going to take place on the 18th to 19th July of 2023. So I thought, sorry, it was a bit, sorry for this. So it will take place in July, and here in Heidelberg, and you can also visit our website and register for this conference. And with this, I would like to close, because obviously this has been an effort that has been driven by a lot of people inside Heidelberg, outside of Heidelberg, a lot of collaboration, a lot of important collaboration, again, showing how important it is to work interdisciplinary in this field. I want to thank all the members of my group who have been driving this work, and the funding agencies for their support, and obviously also you for your attention. And with this, I'm happy to take questions.
So, dear Varun, that was an excellent presentation, and really for me as a neuroscientist, it was very exciting to go through all that. I mean, it's a completely new universe, right, and different view of the cancer itself. So for the moment, I don't see any questions from the audience, so I'll just take the advantage of going ahead So I was just curious, has there been any research done on the effects of the temozolomide, if I'm pronouncing correctly, on this network that has been observed in the tumors? Has any work been done on that? Because it's the only available treatment for the moment, right?
Yes. Yes, this is a good question. And I think this was work done also in Frank Winkler's group, who used the temozolomide treatment to investigate these gap junction coupled networks. And what one could see again, also similar to what was experienced after radiotherapy, that indeed these tumor cells show much more gap junction coupled connections after temozolomide. So again, showing that these tumor cells seem to, or these tumor networks seem to confer primary and adaptive resistance to therapies. Okay,
that's interesting. And in terms of, we have the two populations, right? The connected and the unconnected tumor cells. So do we know during the tumor evolution, do we have first the unconnected that later on evolve to connect? That's I mean, from the neurodevelopmental perspective, that makes sense, obviously. And having the AMPA receptors first and then the NMDRs, because you have the synaptic, but there's no actual synaptic communication, right? They're just active in terms of EPSCs, not actual action potential. If I observe the
yes, exactly, yes, absolutely, yes. And I think this is exactly what we are trying to figure out at the moment, is to really understand how these tumor cells evolve over time. And again, from a very broad perspective, we see that these unconnected tumor cells indeed evolve to connected tumor cells over time. But I think there are much more mechanisms also at play. And again, also, we need to understand this is what we're talking here also about natural brain tumor evolution. So the question is also, how again does it behave after therapeutic interventions, and how does the regrowth happen? So I think these are all unclear questions until now.
Okay, and in the models that you used, you showed at some point the human brain sections. And did you actually observe in the human specimens these two populations, and in the ones that in the model that you use, experimental, that you actually did these observations with the tumor networks and the microtubules? How was the tumor induced to achieve these observations? Was it in a specific context, or does it apply to different causes of glioblastoma?
That's a good point. There were several questions, so I'll start. So just remind me if I forgot something. But so we do see these different types of populations in human tissue. Yes.
Okay.
So we can reproduce all the findings that we have in the xenograft model, and we are also seeing that. When we think about the models that we used both syngeneic models, but so basically mouse cells, but mostly we are working with patient-derived xenograft models. So we use patient-derived xenograft models that we have as a cell line in culture that were transduced with a fluorescent construct and then injected into the brain. So, but again, showing in the several models, we do see the same thing.
Including organoids, for example, or like
organoids seem to be more difficult, in my opinion. So I think the problem with organoids is that you not necessarily have a mature nervous system within the organoids. So it seems to be more reflecting of a neurodevelopmental phenotype. So so people have been doing this work with organoids, but again, I'm skeptical whether this really gives or how well it recapitulates an adult nervous system. I think the clear advantage is that it's human, It's
Yeah.
a pure human system, but again, it's also not very applicable to techniques such as electrophysiology. So, so I think there are, I think this is, and I'm curious to see how this field will develop over the next years.
Yeah, because you need, I mean, in a slice, you can do the electrophysiology, obviously,
Yeah,
but then you have the human challenge, because
sure.
that's a critical factor. Have you actually worked with like diseased brain samples on electrophysiology? Is
Yeah.
that even possible?
Yeah, it's
Okay.
very difficult, but it's possible. And I've been also talking to other people who have been doing this work. So again, we saw in a few patient samples that we were able to acquire, we do see also in patched tumor cells that there's indeed these tumor cells that show EPSCs.
That's very interesting. And what was the other question that I had? So considering that, I mean, the tumor microenvironment, it's a whole different level of itself, a different like component. Beyond having the tumor cells migrating and touching the blood vessels, is there any paracrine interaction that, along with the synaptic, or if you can say it's synaptic activity of the tumor cells that we have observed? Because you have the tumor cells that are kind of push themselves to evolve and induce further tumor cells to be produced. But what happens, how does the blood vessel network come into play along with the neuronal-like activity that we observe?
Yeah, that's a very good question. So I think these are all very unexplored topics so far. So we don't know, basically.
We don't know. Okay, so more to learn.
Absolutely, yes.
Okay, so I have no questions yet. So just for people that are hearing us, if you maybe you wanna give us an overview of the conference that is going to take place this summer.
Sure. So I think these are going to be 2 days of international. We have a lot of internationally invited speakers, and we will try to cover exactly what is state of the art in the field, what do we need, and how can we really achieve that as a community as well. And again, we try to bring together both new people who are already working in the field, but also neuroscientists and also neuro-oncologists, but also oncologists. So basically, because it's not necessarily restricted to to brains with to brain tumors, but it's also an important field in peripheral tumors, so tumors outside the body. So we try to bring all these fields together and try to understand much better and want to learn also from outside disciplines to really inform our further research.
Yeah, I think it's important to have like an out-of-the-box perspective in this situation, because that's what's gonna push forward the field.
Absolutely.
So since having the interest, do you have any collaborations with biobanks, and how do biobanks can participate and help push the field forward? What would be something that you as a doctor and researcher on this specific topic would like to see, or would it be the helpful factor for this kind of research in terms of
that's a very good
biobanking?
question, I think, and I think it's an important question. I think, thinking about biobanks, I think it's I would say my dream would be that we have tissue, because all these 10 techniques that we're just discussing and saying, it is not super easy to do this with frozen tissue or paraffin embedded tissue. So what we would need is to really look at, for example, only either fresh tissue, which is obviously always difficult, but again, from a structural point of view, it would be already great to have freshly fixed tissue that is not embedded in any other kind of material, because this would already give us an important idea how the structures and what kind of neurons, what kind of other cells do play a role. And I think having such larger cohorts of samples would certainly be extremely interesting and important.
Yeah.
So that, and you know, I think you need to basically what I would love to establish, and what we haven't been able to do so far, is to really come up with a more cancer neuroscience adapted biobanking pipeline, which again is because I think people were not necessarily interested in these types of things, so no one was doing it. But I think this is something that I would have in mind.
Yeah. And how about like peripheral markers? Like, can you use in this, like, can you use blood or CSF for like biomarkers? I don't know if it's normal to take CSF.
I think you're absolutely right. I think this is exactly one of the steps in such a program. But you know, the problem is we need to make sure that the markers that we're reading out do tell us something about the phenomenon that we indeed would like to study. So what one would need is a comprehensive characterization of a cohort where you know how tissue looks like, what is going on in tissue, and then correlate this back to the liquid markers, you know, because otherwise
you don't know what you're looking for.
What you are looking for, and whether this really is simply an artifact, or whether there's simply no signal there, indeed.
Exactly. And my last question. So glioblastomas, you see them also in kids. What do we know anything about glioblastomas in kids in terms of neuroscience?
Yes. So this is basically work that Michelle Mongi in Stanford is primarily focused on pediatric gliomas. And again, we do see a lot of parallels between what we see in adult gliomas. We do see also reproduced in pediatric gliomas. So yes, there are certainly parallels, and I think probably there are also some differences.
Yeah, but it's important because if you think about it, in the children, you have a developing organism, right? So you have the extra layer of plasticity, but those developmental mechanisms are hijacked by the glioblastoma even in adult stages. So something has to give, I guess, in this situation. But I guess in the children it's much more complex because you have that extra layer of complexity of the developmental mechanisms. I mean, they're still developing. So that's even more challenging for the treatment of their children.
Absolutely. Yes, I agree with that.
So I won't keep you further, but although I'm very interested in this, obviously, it was a pleasure having you today with us and giving us this very nice and interesting presentation. And we hope to see you at the conference.
Sure. Thank you.
Thank you again. And I wish you all the best in this new adventure that it's beginning in this field of research. Thank you very much, Varun.
Yeah, thank you so much for the invitation. Yes, have a nice day.
Thank you. Have a nice day. Bye-bye.